Hello, I'm Lauren Walker. I'm a senior lecturer in clinical pharmacology and therapeutics and associate director of an NIHR phase 1 clinical research facility in Liverpool. I've been asked to prepare a talk today on something that's very important to me, which is about inclusivity in clinical trials, and in particular thinking about female biology and the need to include females in early phase drug development. So why it matters, and it's increasingly acknowledged that clinical trials for new therapeutics haven't really accounted for sex-specific differences in the data that they generate. And that means that the data that's used to determine safe and the optimal dose for any given new drug has been largely done in a data set that excludes 50% of the population. And it's important because pharmacokinetics, which is what the body does to the drug, and includes how it is absorbed, how it's distributed throughout the tissues, how it's metabolized, so how it's changed, and how it's eliminated, how it's removed from the body, is often completely different in females than males. Yet there's a lack of safety data in females, which is owing to their absence really from early phase clinical trials, and there are a number of reasons for that. But it's those trials that are used to determine the safety parameters for the drug. Not for, when I think there's a greater focus on inclusivity in later phase trials when we're thinking about whether or not the drug works, but I think it's particularly important when it comes to having that optimal dose. And the differences between the sexes can affect both the pharmacokinetic and the pharmacodynamic parameters. And largely those differences often resolve around weight differences, differences in muscle mass, in lean muscle mass, body fat distribution, the metabolic enzymes present within the liver, and plasma protein, particularly relevant for drugs that are highly protein bound. So if you take the example, one example would be that females typically have a lower body weight than males, not always, but often they have, for healthy females, a lower body weight than males. So when taking the same dose of a drug, that can result in a higher level of the drug within the female system. And one example of that is a drug that's used to assist in insomnia, so in a difficulty to sleep, so the drug Zolpidem. And the US FDA issued specific guidance to industry back in 2018 to lower the recommended dose of Zolpidem from 10 milligrams to 5 milligrams for females. So when prescribing in females, the recommended dose is half that of the males, and that's following a study that looked at next day impairment. So that was for when they looked at the blood concentration of the drug the following morning, it was significantly elevated in females, which can impair things like driving, the ability to drive, or the ability to function in work and things like that. So that's one of the only drugs I'm aware of that has a specific female dosing recommendation. And then there are other examples, highly lipophilic drugs, so drugs that distribute into fat rapidly. They tend to have a larger distribution in females because of their higher body fat content. But it's important to note that the difference in sex contributes to not only anatomical and physiological differences, but also has wider influences on behaviour and disease risk. The risk of acquiring certain diseases is different in males and females. And there's a historic underrepresentation of females in early phase clinical trials, and that means that we now lack the knowledge around the safety of drugs when they're prescribed to females, and therefore they often experience more adverse events as a result, which is in itself a health inequality. And a good example of that is in the area of cardiovascular drug for cardiovascular disease drug development, which was largely undertaken in middle-aged males, and then that data was extrapolated and used for dosing in everybody. But we know that receptors for the sex hormones, so for the estrogen receptors, progesterone receptors, they mediate sex-specific effects on the cardiovascular system. So your alpha and beta estrogen receptors, which modulate myocardial gene expression, hypertrophic and apoptotic signaling, your ion channel expression, which is relevant to drugs that might prolong might cause ECG abnormalities. And also that vascular estrogen receptors exert vasoprotective effects, which reflects on the difference in cardiovascular disease risk in males and females. There was a review that was undertaken of cardiovascular disease drug development trials that showed for studies of angiotensin converting enzyme inhibitors, which are widely used, beta blockers, aspirin, digoxin, and antiarrhythmic drugs, that females were significantly more likely, and that's about 1.5-fold more likely to experience an adverse event. And then that was particularly true for drugs that prolong the QT interval, so that can cause electrophysiological disturbance in females, and therefore they were more likely to experience life-threatening arrhythmia. And that in itself is represents a health inequality, that females are taking medicines that were not designed with their biology specifically in mind, and therefore they're at a greater risk of the adverse outcomes of those medications. And if you look at aspirin, widely used, the bioavailability and plasma levels of aspirin are higher in females than males. And that's because they have lower clearance and lower activity of aspirin esterase within their body. So females have more severe bleeding than males, attributable to aspirin. And then there are differences in terms of the disease expression in itself. Most autoimmune diseases are more common in women. Hypertension in young people is more common in men than women. So, when high blood pressure is diagnosed earlier in life, it's far more likely to affect males than females, owing to some of those sex-specific differences. And diabetes and hypertension are more severe risk factors for cardiovascular disease when they occur in females than in males. So there's an important historical perspective to this, which largely follows the thalidomide tragedy in Europe. And the concerns about potential reproductive adverse effects led to policies and guidelines that considered pregnant women as a vulnerable population. And so they were then excluded from research trials, and it restricted the ability of women of childbearing potential to even enroll in trials, and especially for early phase stages of research. And it's easy to feel that that there is an inherent risk there to two individuals. That if a woman of childbearing potential were to get pregnant, and you're increasing the risk. And I can understand there is a natural tendency to avoid that risk. In the US in 1977, the US FDA effectively banned females of childbearing potential from taking part in phase 1 or phase 2 trials, unless it was for drugs that were life-saving interventions, and then they were. And it was sort of defined very widely as any female capable of becoming pregnant. That's how childbearing potential was defined. And that included those who were completely abstinent, people who were on hormonal contraceptives. It even excluded those that were in a relationship with a sterilised partner, so a male that had a vasectomy. And that view is largely now seen as being overly paternalistic, and leaves no room really for the exercise of judgment, either by the participant or by the supervising physician. By banning it, you're saying that you have no ability to control for or understand and accept, that risk and make that decision for yourself, which isn't. Although I can understand the tendency, it's how we've ended up with a wide therapeutic arsenal of medicines that can cause quite significant risks for females. And following that sort of, for about 20 years that persisted, but there was increasing concern that the guidance caused underrepresentation of females in drug development studies, which is true. And the FDA then reversed that guidance in 1993 and lifted that ban, and it left the decision, through the informed consent process with researchers, independent ethics boards, and females themselves, which seems quite right, really. They went on to articulate a need for including trials the intended population, the recipients of the drug, and so that of course includes both sexes. And then that was further developed about 5 years later when it became mandatory to examine data appropriately by sex difference, and that's the view that the NIHR takes in the UK. The Association of British Pharmaceutical Industry, the ABPI, recommends that a woman capable of having a child may take part in a trial of an investigational medicinal product if the reproductive tox studies have been completed, and so the results raise no concern against participation, and or the risk of pregnancy is minimised because the female agrees to use a highly effective method of contraception that results in a low failure risk. and obviously that she's not actively pregnant at that time, warned about the potential risks to a developing child should she become pregnant, and is tested for pregnancy throughout the trial. And that's what we do. That's what we do within our unit. And most, pretty much all, the NIHR would recommend the use of not only a highly effective method of contraception, but a secondary method of contraception as well. And therefore, that mitigates that risk. Doesn't exclude it entirely, but it mitigates that risk. And I think more and more we're realising that the differences in drug processing between males and females are dangerously understudied, and that pervasively and fundamentally that sex difference, that physiological difference, it does matter. And therefore, females should be prospectively included and evaluated throughout all phases of drug development. Thank you.